Hello everyone. Yes, I think I am online again. I hope that you are able to hear me and see me. Let me quickly get everything sorted out. Okay, maybe you can give me a short thumbs up if you're able, if the audio works. Because as you probably noticed, I am now streaming from a second microscopy channel. Yeah, thumbs up, I see. Thank you very much. What I've done is I've reorganized a few things on my YouTube channels. Okay, very nice. I would like to simply share with you a couple of things that I've now reorganizing a little bit. Today's live stream, I've actually prepared a couple of topics here, and I've now moved this live stream back to the Microbehunter microscopy channel because I'm going to now use this channel here for all microscopy related topics. So I've prepared a few here. And the second thing that I'm in my main Microbehunter channel, what I will be doing from now on is I will be putting out some biology, microbiology related videos where I'm going to be explaining the world of microorganisms and microbes. So now that I got that straight. So in other words, I'm currently reorganizing everything a little bit. What I'm going to do now is just to give you a quick overview. I've actually prepared now a couple of topics here that I would like to cover today. For each one of those topics, I'm going to spend approximately 15 minutes, and I'm right now in the first 15 minutes up here, right? If you go to the very beginning of this chat, then you're going to see a link with a poll, okay? And if you want to, you can vote on what you want me to put under the microscope today. I've got two possibilities. Their link is the first in the first comment of the chat. I've got two specimens here. This one here is I just found at the supermarket. It's some kind of strawberry, banana, apple, mashed something, biological stuff here to put under the microscope. Or this one over here is vegan meat. I think it's some kind of a soybean type of thing, and it's also quite interesting. I already put it, of course, under the microscope before. There is something in there that I also wanted to show you. I've actually got several of those. It's all in alcohol. Right, that's why it's able to store. Or we can look at a couple of biological samples in here. Some of you requested that we do both. No, I'm only going to do one, but don't worry. I'm going to next time again put a poll on so that you can vote again. So if you want to, you can click on the link in the chat right at the beginning, and then you can vote which one you want me to put under the microscope. I see that currently, 31 people have already voted. Currently, 61% of the people voted for the vegan meat. We're gonna see, okay? So what I'm going to do now also is I'm going to just say hello all around the world, okay? From Vietnam, from Belgium, from England, from Wales, from France, the United States, okay? Very nice. Essex, UK, Switzerland, Ireland. Okay. So the thing is the following. There's already a comment here that I would like to talk about here. Is this the reason why? We're going to see how this works out. Yeah. So I'm going to in my other channel. What I would like to do is I would like to explore a little bit the world of microorganisms and such. So I'm going to stay true, of course, to the main format in the sense that it will be about microorganisms. I will be, of course, also sharing microscopy related material. But I thought that all of those specimen preparation things, and when I talk about the technology and microscopes as such, that I'm going to move this this channel here. I have to be honest with you that I've had problems with my other channel. Views were going down over the last couple of months as well, and I think it's also one of the reasons because the formats were not quite clear. So I'm going to try it like this. So basically, the idea is it makes it in the past. I've split the channels like this. I have had the Microbehunter microscopy one, this one over here, to talk about optics, for example, and microscope hardware and microscope reviews and so on. And the other one I did for specimen preparation. And the thing is that what I want to do now, however, is I want to separate this a little bit, because this channel here is going to be a little bit more specific. So I will also talk today about some optics here. Right. This is going to be the channel now, which is going to be a little bit more the hobby microscopy channel. And the other one I want to keep a little bit more general, where I talk more about, for example, I've prepared now a video script. I'm going to share it with you. The script that one of the videos that I'm preparing right now for the other channel would be why it is important to disinfect space probes. So, for example, the Cassini space probe several years ago, they crashed into Saturn to burn it up to prevent microorganisms from possibly contaminating some of the other planets and moons there. So you see, it's still microbiology related, but it is not so much microscopy related. So this is a little bit, the idea is the following, that what I will be doing is I will be, for right now, I want to keep the live streams which are microscopy related on this channel now. 2 years ago, I basically did live streams here, then switched over to the other channel. Now I'm kind of switching back a little bit, but otherwise it's going to be the hands-on microscopy stuff that I've been doing up to this point as well. Okay, so this is a little bit the thing that I'm going to try right now. So it will not be ever smaller commented. Biology lesson is great, but I'm not sure if it's the best move. Most expect general microscopy on the main channel, and that's where your biggest audience is. It is correct that my main channel has the biggest audience. However, relatively few of my subscribers actually watch the videos. So this is a little bit, I have to also admit, I've got slightly the wrong subscribers. And one of the ironic things, the last video that I posted on my main channel is actually where I talked about me stopping to make YouTube shorts. Where basically YouTube shorts kind of destroyed my channel. This was actually a completely non-microscopy topic, and this video got actually more views than some of my microscopy related videos, which is kind of ironic a little bit. So this is a little bit the thing that I've been experiencing. We're going to see how this works. Okay. So hello from Athens, from Osnabrück in Germany. Okay. From New Zealand. Again, a comment, please make sure whichever channel is on each week to be sure to post on the opposite channel. Yes, I will try to do this as well. I want to give you quickly an overview here of the ideas of what I intend to cover a little bit this time. I want to split up this live stream also into separate chapters. In the past, I had more or less one main topic, and then I kind of rambled along a little bit, and then I tried to cover approximately 1.5 hours with questions at the end. And sometimes this was a little bit difficult for me because I had to kind of, I didn't always have, not all of the things that I wanted to show you actually were interesting things to be seen. So what I've done is the following. I would like to, currently I'm still in the editorial for 15 minutes, right? Then I would like to show you, and we're going to make a slide with trichomes, which for 15 minutes. So I'm going to make a permanent slide here because I think they look kind of nice. Then I would like to answer a question about soil microscopy. I got 3 questions here about soil microscopy that I would like to talk about, and I actually did also prepare a soil sample here that we can look at. Then I thought about trying out to do a little bit on microscope optics as well, because I do want to show you 2 different types of objectives that I have got here. And then specimen 2 would be the viewer's choice, the one that you voted on, where essentially it's either the vegan meat or the mashed fruit, and currently we've got 65 to 35%. Yeah, and then at the end again, I would like to do some Q&A. So if you have any questions that you want to post in the chat, please indicate this at Microbehunter or at Oliver so that I know it's for me. And then at the end, I would like to answer those questions. Okay. Yeah, so I look forward to seeing how this goes. Though I'm here for the ride in either way. Yes, I also am a little bit. I have to tell you, it is also a little bit of an experiment. So that's something that I simply also want to share. And I'm going to stick to the 15 minutes. Very briefly, also something that I would like to show you is something over here. This is, where is this? Where did I put this? Ah, yeah. So that's actually also something that we might, not today, but I'm also going to put this into the poll maybe for next week. This is some snake skin. Okay, that we found in the forest. So, you know, the snakes, they actually shed their skin. It's a protein, it's called keratin. And these specimens are also quite suitable to do, how do you say, for stereo microscopy. So I also wanted to do that a little bit. So please, every week also check the polls. Yeah, I would like to again then also next week make a poll so that you can choose which specimens you want me to put under the microscope. Okay, so this is a little bit the idea. So another thing that I want to briefly mention here is the following: a little bit of a recap, also from last week. Yeah, you see those slides here, right? Maybe if you remember, I made those ringed slides with glycerin gelatin. Okay, not very nice looking, but I simply wanted to show you that when the nail polish starts to dry, of course, it starts to shrink, and now then we can reapply another ring here, right? So, yeah, by the way, before I forget, if you check now this channel here, you might notice that in the over the past couple of days, I've uploaded a video once every day even. Okay, so what I'm trying to do now here is I'm also going to try to go up a little bit with the upload frequency, and the videos are going to be completely unscripted and pretty, what would you say, kind of raw. Yeah, simply where I'm just communicating with the community a little bit and just sharing my thoughts. So that's also a little bit part of the experiment, a little bit to see how this actually goes. Yeah, yeah. So I'm just checking a little bit. Yeah, I look forward seeing how this goes. Okay, so what I'm going to, there have also been some requests. There have also been some requests about changing the time. I know that people around the world watch this live stream. I mean, you can also watch it afterwards. It's going to be online on YouTube in any case. But for me, it's the most convenient time, unfortunately, right? Because sometimes I do have evening appointments as well. And currently, when I do the live stream on a Saturday evening, it's 9:30 in the evening, and that is the time when I'm most of the time I'm actually at home. Yeah, so that is a little bit something that I just wanted to share with you. Okay, so you know what? Let's slowly get started with this one over here, Trichomes. Just want to show you, what I got a couple of maybe already 2 years ago. Somebody sent them to me. Some leaves and for me to put under the microscope, and indeed there was a little bit of a fuzzy layer on the surface of the leaf, and these leaves are basically so-called trichomes, which are plant hair, and they look kind of pretty, and I thought, well, why not prepare it again? I've already done this before, so sometimes I do reuse the samples and specimens that I show you, but they do look kind of nice, and for this reason I decided to actually also make a permanent slide using this here, and then of course we're going to look at it together, right? And because I want to also keep it. Look, I've also made a slide label. Yeah, so so later on I'm also going to glue this on here. Let's do it like this. Okay. So what I'm going to do is because many people told me that they had some problems getting appropriate mounting media. So what I'll be doing now, therefore, as a general purpose mounting medium, is this Elmer's PVA glue, which I already mentioned. So just in case you have not seen this yet, it's this glue here. If you're in the German-speaking world, or especially in Europe, there is this type of glue that we've got here, and this one is also so both of them are water-based glues, and this one over here is also water-based. So it will actually say in the back here, that's free of solvents. Yeah. Well, actually, the solvent is water. Yeah, what they actually mean is it's free of organic solvents. And so it's a water-based glue, just like this one, and therefore it's also quite suitable. And I've also had reasonably good results with those, right? So the first thing that we have to do is if we want to collect the trichomes here, is we have to kind of collect them from the surface here. And one way of doing that is carefully trying to scratch the surface. And then there is this fuzzy thing here that you might see, which is a little bit difficult to see, and that's these are the trichomes. And what I'm going to do now is, so not all leaves have those. Unfortunately, I don't know from which plant they come from. Yeah, that's a little bit of a pity, but yeah, I'm going to use now the glue and then we're going to simply look at those and enjoy what we see. Yeah. Actually, you know what? I have to be honest with you. I need to do a preparation step here. So let me get a pen. I nearly forgot that because I want to make sure that everything is properly centered. So look, then I'm going to just use this piece of paper here. And what I'm just going to rip this paper here. Okay, so because I do want to make now a permanent slide that basically hopefully will last a little bit longer. Yeah, I should not have touched it because now it's greasy. And going to simply make mark the center approximately here. Yeah, sometimes it's going to drift anyway a little bit. So I think that's much better like this. So where is the glue? Now we can actually start to apply a drop here. Again, bubbles are always a little bit of an issue. And the next thing is now we're going to put in those trichomes, and I would like to take now a toothpick. Let's carefully try to, I don't know if you're able to see that. And what sometimes is a good idea is to try to kind of spread them apart a little bit, because those trichomes, which are planted here, they sometimes like to stick together, right? So that's hopefully good enough. Yeah, I don't know how many there are. Okay, yeah, there are gonna be a few. You know what? Maybe we're gonna add a few more. This fuzzy. Yep. Here it is. Yeah, do you see this fuzz? And these are the trichomes. Make sure that they're properly submerged. It might be also an idea a little bit maybe to moisten them first a little bit in some water. Maybe to drive out any air that might be present. Just gonna do it in any case like this. Cover glass goes on top. Those cover glasses luckily are reasonably clean. Goes on top. You can let capillary action do a little bit, some of the work. And yeah, we're gonna press it flat. Usually it's going to spread a little bit. You have to be careful that it doesn't, not too much of the glue spills out. If it does, then you can always remove it. And I decided that when we already make slides, that we make them properly. And I would like to add a little bit of glue here as well. And spread it up. Okay. So there's a question already, what do the trichomes do? Yes, the trichomes, they can help with water retention, but they can also serve as protection against insects. Okay, because those trichomes, they reduce the so-called transpiration, because on the bottom of the leaf there are the holes called the stoma, and over the stoma, of course, oxygen is given off, and carbon dioxide is taken into the leaf, but also water vapor is lost. And if there's not this fuzz on the bottom side of the leaf, then basically the diffusion of water vapor is also reduced. So those plant here sometimes have multiple functions, and let's have a look now at what we have here. And and I'm going to try to stick a little bit to the time scale that we have, to the time frame that I have. So this means that I just started with trichome, so that's about so I'll spend approximately a total time of 15 minutes here. So let's give it a look. And this is, I have to always change around the. Let me see. Is the here we go. Okay. You see, it takes a little bit some time until the cameras start to, So, and here they are, okay? Sometimes you do find them in water samples even, and so these are basically those plant here. When I saw them several years ago for the first time, I said, what in the world are those? They look a little bit like spiders. I see. That's why they kind of are clustered together over here, right? That's why it's a good idea to kind of spread them apart. And what we have here, that's actually one of those, unfortunately, one of those, under quotation marks, problems. And that is if they're too much clustered together. You see over here, the dark line here, that's of course an air bubble. So the glue is not, does not completely cover them. I think it's okay. But just to have turn off the arrow again. So, and as the glue dries, of course, what I've seen, luckily with this glue is that it does not shrink too much. Could you please remind us what they are? So yes, those trichomes are hair structures that can be found on the surface of plants. So like, for example, this plant leaf, and they have multiple functions. And one of the functions is that apparently that they protect the plant also from insects, because if they're tiny little insects or maybe even mites, they might have a problem actually reaching the cells of the leaf, because there's this fuzzy layer of those trichomes in between, right? So that's so it's a protection apparently. Because some of those insects or mites might actually feed on the sap, on the juice of the leaves. They want to bite the leaves maybe, and if there's this fuzzy layer on there, then they're not able to actually reach the cells. So that could be one reason. And another reason is very common in plants, what you have. And you know what? I'm going to actually make a little drawing here to illustrate this. If you have now, I mean, this is not in a biology lesson, but I'm going to draw the lower. If this here is the leaf, right? And over here, this is what we call the lower epidermis, and what we've got here is we've got so-called guard cells, which are basically, and that's a stoma, which is an opening, and we've got here again cells, and then this is the leaf, and then there is the problem that essentially water, H2O, water vapor is lost during the day, right? And the plants lose a lot of water. So in many plants, what they have is they have those plant here, those trichomes growing here on the bottom, of the surface, and this reduces the water loss because the water cannot diffuse out as easily here. And this fuzzy layer, those trichomes, those plant here, of course, as I just mentioned, they also kind of prevent, if there's an insect over here, right, then it also has a problem reaching the actual plant cells. So it's not able to drink the sap of those plant cells, right? So that's apparently so usually, as you can see in biology, some structures actually have multiple functions, and those trichomes basically they're growing here, and they can have different shapes and forms, So what I kind of like about those is that they do look quite nice, Where are they? Just a second. It takes some time again for the camera to switch on. Here we are, right? So let's go up with the magnification. Let's pick out one of those here. I don't know. Here they're clustered together. I mean, maybe over here. Okay, and yeah, let's go up. In case you're again, maybe if you're new to the channel and if you're wondering about the blue background, that's because of my optics. Normally, if you see them in bright field, this is how you would see them normally. So these are the natural colors here. Yeah. So these are the natural colors. Yeah. So, and we go up, it is now 40 times. Yeah. Yeah. And you can, let's close the condenser is already closed. Yeah. Yeah. So I think they, yeah, they look quite interesting and pretty. Yeah. So again, over here with the, yeah. So what we can try to do, however, and this is also something that only works with the low, and I did not try this yet, okay? I did not try this yet. So this is now the no polarization. So this is now the 4 times. And what I'm going to do now is I'm going to place, you might want to check what's happening, going on over here. I'm now going to place a polarization filter. To remove the other one over here. I'm going to on the bottom, and I'm going to place also one on the top. And of course, know that I'm going to be touching the cover glass now. And let's see if these are somehow polarizing or not, but they don't seem to be. Maybe they are. Okay. Let's see if they're... Yep. Okay. So this is now basically in cross-polarized light. Yeah. So there are also plenty of opportunities. So if you want to try this out yourself, cross-polarized polarization, then that's quite easy to do, because all you have to do is you have to place, and I'm going to show this to you as well. Let's go to the desk view. Let's put this away. Yeah, and let's If you're also interested in doing polarization work, all you do is you put a here this is a glass filter, but you can also buy a polarization film, one on the bottom, one on the top, and you put it into crossed position, and then you right now there is no light from the bottom, but then you can also see them shine up on a dark background. Yeah. Can we see the plant where the plant creates these trichomes? Yes. I'm going to show you again the leaf. Okay. Over here it is. Unfortunately, I don't know the name of the plant. Okay. Yeah. Also, microscopy enthusiasts sent them to me. Yeah. So they're right now they're dry. This is he said this plant was growing in his garden. Yeah. And if you actually look on the bottom, then you can, I don't know, you know what I'm going to try? They're difficult to see, okay? So what I'm going to do now is let's see if we're able to find it with a stereo microscope. Let me see if this, yeah. Okay, I'm going to put it under the stereo microscope. But you see they're a little bit, yeah, the contrast is too low. Yeah. But I'm going to try the following. Let me see if I do that. Maybe you're able to see that there's this fuzzy thing, right? Yeah, this is fuzzy. Yeah, you can actually see it much better now. Let me focus again. Yeah, again, the contrast is not the best, but you see that there's this on the surface of the leaf. There's this fuzzy layer. Yeah. And so it's quite dense, yeah, and that's basically the thing that the plant is kind of creating here, yeah. So just wanted to show that to you, okay, and that is basically something that I simply taken and put on a microscope slide, and where is it? Here it is. Okay, here it is again. I have Yep, that's a regular bright field again. And if you want to see it a little bit with color, then we have to switch on the other filters here. Here we go. Yeah. So I'm just reading again. Yeah. There is a from Dar Wexter. There's a question for me, which I'm going to actually want to answer those questions, but very quickly. Yes, I tried the 3D with the polarization. I'm still working on it and optimizing it because I did not get a very strong 3D effect. Okay, just very briefly to answer this. Yeah, I think it must have been the bottom of the leaf. Yep. And the top one is over here. Yeah. So so that is kind of the how do you say the thing that I wanted to show you. Okay, and I'm now going to move on to the next topic because I'm trying to divide this a little bit into some chapters. So I have to do now the following. I'm going to put the leaf back again here into the bag. So this is a kind of a little bit of an idea. One of the reasons why I'm also doing these videos and the live streams is also kind of so that other people who are into microscopy get a little bit motivated in trying out some of the new things. The next thing is the following. I want to talk, so you see I'm jumping around a little bit with the topic soil microscopy. Why in the world that? Because I did receive a couple of questions that I want to show you. Per email and also on the YouTube, and for whatever reason, surprisingly many people seem to be interested in soil microscopy. However, they're not always happy with the results here. So this is an email that I got here. I hope you don't mind if I ask you one question here. So I already responded to this email, of course. But I wanted to share this with you because actually I think it's still quite interesting. And now I have my microscope and I'm of course enjoying looking at things. However, I'm finding it difficult to find any microbes in soil samples. So far I have found 2 nematodes and a couple of dead microbes. Now, being that this is over the last weekend and where I live in England, temperatures hit over 30 degrees, I'm wondering if heat and dryness of the soil is the reason I've been struggling to find anything. Short answer is yes, but not only. Okay, any help that you can give will be very much appreciated. So, and then the second question is the following. Does it need a special microscope to view soil-based microbes? I see hundreds of videos of microbes in water, but hardly any of ones in soil. Is the technical reason for this? No, not a technical reason. I will also talk about that. The reason is because there are different kind of microorganisms in soil, and they don't swim around as much. And I'm interested in soil microbiology. This is a huge topic and I'm a beginner. Would you explain what to look for in a rich, healthy soil compared to poor unhealthy soil? So this is kind of, I would say, an interesting topic here. And what I wanted to do is the following. It's just, why not simply have first a look at a soil sample? And the reason, but let me also answer it, the reason why there seem to be fewer microorganisms in soil is because there are a lot of fungi and bacteria in soil, but they don't always move around. Okay, if you look at a water sample, then because it's water, it's in a liquid, you actually see more of them because they are moving around actively. So that is actually one of the reasons. And of course, in soil, you have different types of microorganisms. You have fungi, for example, that don't move around as much. Yeah, so that is one of the reasons. And for this reason, I said, okay, why not just put a soil sample also under the microscope? Because I also want to demonstrate something to you, for you. Something that I'm kind of, or maybe I can use the other slide over here. It doesn't really matter. Yeah, I'm recycling those slides in any case. So what we have here, the big problem with any sample like this is, look, lots of particulate matter and difficult to pick up sometimes. So what I'm usually doing is the following. I take a pipette. You can also use disposable pipettes. Yeah. And either if you use this one, then it might get clogged. So sometimes I simply cut off the tip. And now we of course have the problem that there might be a lot of solid material. No, actually not now, not so much. But I'm going to give you an example here. If let's say there is too much solid material on here. I'm just going to show this to you quickly as a demonstration. Okay, look, I'm just going to show it to you here. Lots of, So if I were to place a if I were to place a microscope cover glass on here, it's not going to be flat because there is simply too much thick solid material on here. So my suggestion is always the following: is either you kind of only pick up the liquid, or you can try the following. You just take a cover glass, you kind of just push it to the side like this, and go like that. Yeah. Then whatever is in here, and the rest over here, because there's always the danger that you're probably going to get your objective wet. What I usually do is the following. I'm simply going to take some tissue paper to remove any excess. Okay, that's kind of what, And then we're going to simply have a look of what we're able to find here. The reason is if we leave those soil particles, then there's so much of that any microorganisms might be covered up anyway by those. So let's put this on here and let's have a look at what we're able to see, and I will tell you probably not very much. Okay? So don't be disappointed. Again, I'm waiting for this thing to switch on, and that's basically let's go up a little bit here. That's what I'm able to see here, right? Yeah, whole bunch of up there. Sometimes there is a little bit of movement going on here, right? But it's actually no comparison to a water sample. I mean, if you remember what I've last week. I mean, there is a bit of movement going on, and however, the thing is now, does this mean there is actually nothing living on here so much? Well, of course not, because it's full of fungi and spores and bacteria, but not all of them actually move. And in your comment over here that you actually mentioned, you basically have seen some dead ones as well. Yeah, under the microscope, you don't really know if an organism is dead or not. Right, just because it's not moving does not mean that it's actually dead, right? So this means that sometimes it's simply difficult to see. So what I usually do then is switch over to phase contrast. Okay. Okay, well, at least a little bit of movement going on, but in phase contrast, what we're now able to see in the background here. And that is actually the thing that I wanted to make clear is many of those tiny dots that you see in the background are bacteria, and they can be extremely difficult to actually see under bright field. So what I'm trying to say here is and that's maybe actually one of the questions over here is do you need specific equipment? And I'm saying, well, I mean, if you want to see bacteria better, you're better off with phase contrast because it's just easier to see. Right, but it does not. I mean, it's absolutely necessary. But the point that I'm trying to make is that obviously a soil sample looks very different from a water sample. So this is So what I've also done is the following. This here, I added, I don't know, maybe an hour or 2 hours ago, I added some water to it to allow the microorganisms to regenerate a little bit. And the soil was not completely dried either. So if you actually completely dry the soil, then of course many microorganisms are going to die off. But those that are able to form spores are able to survive. Yeah. So this could be indeed a case. Yeah. Would the bacteria stand out in bright field when staining with methylene blue? And the answer is yes. I like to use crystal violet as well. However, this important is the following is because these stains are so unspecific. They stain pretty much everything. Sometimes it's difficult to still see if these are bacteria or not. Right? So that's a little bit a problem with staining. So let's go bright field. Okay, let's go up with bright field. And then you can actually compare a little bit, yeah. You see there are lots of particles, yeah, and it's difficult to see which 68 times. So it's difficult to see a little bit is, which ones are bacteria. So here a little bit. I mean, if you look very carefully, if some bacteria move in a very directional manner, non-random, but look at this tiny one over here. I don't know if you're able to see this. So then you know it's a bacterium that's able to move on its own, right? And you see there's some other stuff floating around here. So, but it is difficult to see. If you don't have a microscope with a good contrast. So the question, however, is the following: do you need? So the next question is: does it need a special microscope? And I'm saying, well, ideally, of course, if you want to see bacteria, then it's you probably want to use phase contrast, or you want to, as we said, do some staining. And the last question is the following: it's a huge topic, and I'm a beginner. Would you explain what to look for in a rich, healthy soil compared to poor, unhealthy soil? Okay. I'm doing a little bit of gardening, okay? I am doing a little bit of gardening together with my kids and so on. And when we grow tomatoes and other plants, we, of course, want to use soil that is healthy because we're going to be eating the tomatoes that we grow. And essentially, I am not really so much concerned about whether there are many or few microorganisms in the soil. I'm primarily concerned about the following. Number one, I do not like to have any heavy metals, cadmium, lead, whatever, in the soil. That's number one, right? So that's why when we're doing gardening, we're always using properly composted soil from a company. I do not want to have any organic hydrocarbons in there from like oil from a car or whatever. Yeah. So if there are cars parking there and there's oil drip, I don't like that either. And number three, what I don't like is I do not want to have any soil that's contaminated by sewage, because there are bacteria on there that, let's say, or at least human sewage, there are bacteria on there and that they can cause diseases, right? So that is actually what I'm mostly concerned, because the microorganisms, they will come and go, right? If you've got good soil and you're growing, then, and if it's sufficiently aerated and so on, they're gonna grow, right? So that is not so much the issue. I once composting. I once talked to somebody who was doing commercial composting, and it's actually quite interesting. Because when you're actually collecting all of this garbage, under quotation marks, the grass that you want to compost, and maybe all of the stuff that you're kind of dumping together, and when it's composted, what's going to happen is that there's going to be an explosion in microorganisms, and they're breaking down all of the material, all of the waste, and because of this explosion, the growth of the microorganisms, the heat will dramatically increase. So during the composting process, it's really going to heat up a lot, the compost. And this will actually then also kill off ultimately many bacteria. So actually what you have is in healthy soil, you've actually got less bacteria or fewer bacteria than you probably had before during the actual composting process. Yeah, so that is the thing. Super chat disabled. I don't know. I don't know about that. I have to look at that. Okay, because I've changed now the channels, and so maybe it's disabled right now. Yeah, so I have to look into that. Yeah. Austria and Australia, yes, yeah. So that is simply also something I wanted to clarify here. So if you want to observe soil, make sure that it is sufficiently moist, so that microorganisms are given some time to actually reproduce and grow, and then make sure that the slide is also sufficiently flat. Okay, and then you're going to see a lot of bacteria, hopefully. I do remember a couple of years ago, a composting company sent me several different types of compost, because they were kind of separating the compost, and they were making several different commercially composts for different types of purposes. And for their website or for videos, they requested if I could make some pictures for them. And of course, they wanted to see some microbes. Of course, but there was nothing really interesting in there, because you don't have the same microorganisms like the huge, I don't know, paramecia, whatever, that you have in water samples. So in order to provide a sufficiently high or interesting pictures, what I've done is I've actually enriched them so that I could actually show something to them, and then I could take the pictures. Yeah. Are there microbiological indicators for heavy metals that we might be able to see with a microscope? Microbiological indicators. That's difficult. Not, let's put it this way, not that I know of. When you refer to microbiological indicators, basically that we're able to see them on the types of cells that are present. Normally, those metals can be fairly easily detected by chemical means. Okay, and I know that for example, this maybe not so much of an issue nowadays, but it used to be a problem when they still had gasoline, which was leaded. So, for example, it was actually recommended not to eat too many vegetables or fruits which were growing next to a heavily road with heavy traffic, because there was actually quite a bit of lead in the soil, right? But I don't know if there are actually microbiological indicators as such. Heavy metals are like cadmium, lead, and so on. Are a problem because what they do is they are blocking certain enzymes. And therefore they're actually also harming microbial life. But there are always microorganisms that are still able to survive that, right? So it's not a form of disinfection as such, right? So what I'm going to do now is I'm going to move on. Yeah, I'm trying to actually stick to the schedule, and we're going to see how this format actually works. You can of course leave your also your comments. Yeah, even if it appears to be a little bit rushed sometimes. I kind of told myself I would like to try out the new format here. The next one is conjugate. Again, a complete change in topics. And I thought that every now and then maybe I'm also going to talk a little bit about microscope hardware and optics, because some people also would like to know something about that. So for 15 minutes, I'm going to talk about this. But let me quickly check what the viewer's choice is. So if you just joined in, at the very beginning of the chat, there is a link where you can vote which sample you want me to put under the microscope. Currently, the vegan meat is winning with 64%. Okay. So let me quickly talk for 15 minutes or so about this here, because I want to show you a couple of optics, because I've been pulling out my old Olympus microscope again. Just want to show you something. I need to clean up everything here. I've got a couple of microscope objectives here. And then please do in the comments or so, please give me feedback how you like it or not like it, this new format. I know it might be a little bit extreme, changing topics so quickly, but I was also thinking about doing a little bit like a magazine format, because not everyone's always interested in all of the topics I talk about. Okay, and there is another microscope I want to show you. There's a reason why I look this. Maybe I show it to you. It's an introductory microscope. It's a Swift. I forgot the number. I think it's Swift SW150. It's a slightly better one of those introductory ones, because the optics are a little bit larger than the others, and I would like to actually talk about those optics here. And in order to do that, because I don't always want to hold up the microscope, I'm going to simply do the following. I'm just going to take one of those objectives out here, okay, simply to show it to you as well, because I would like to compare them. This here is that's the one that I just removed from the introductory microscope. It has 160 written on here. Okay. This one over here is an old Olympus objective from the 1970s. So also 40 times. It does not have the 160 written on here, but it is a 160 millimeter tube length objective. Look, almost the same size. Almost the same size. And over here, I've got another 160 millimeter objective. Okay, that's basically a phase contrast objective. You see this ring in here? It's a phase contrast. It doesn't matter. It's a 40 times. Look, much bigger. As a matter of fact, approximately a centimeter bigger. And over here, this one is also a 161. It's also a little bit smaller here. Yeah. Because it's a 10 times, but it's also bigger. So the question is now the following. Let's just compare those. All of them have the same magnification. What in the world's going on here? Why do they have a different size? And can I now exchange them? Okay, all of them have a tube length of 160. What does this mean? This basically means the following. Let me take this out here again. I'm going to hold it up here. 160 means that from here all the way to the place where it creates an image in here is 160 millimeters. So basically it creates an image and projects a so-called an intermediate image in here. Yeah, it's 160 millimeters. The question that I have now is this possible now, because all of them have 160, to kind of exchange them? I want to show this to you, and that's why you have to be a little bit careful when you buy exchangeable microscope objectives. Well, actually I can't, because I cannot flip it. Well, it's now upside down, but I hope you don't mind. And there is a problem, or not. I'm going to show you. I'm going to take this here, and I'm going to take now the big objective here. The big one. Let me I show you. And you know what? Here, now you see it. The big one, it fits. Can you believe it? Isn't this nice? Isn't this cool? Yeah, I can exchange it. Yeah, this one here was the original, right? And now I'm able to on this little introductory microscope, I'm able to insert a or not. Be careful. Yeah, no, it doesn't work. And the reason is the following. I'm going to hold it up again. Is this because it's too large? Yeah, you see? Yeah, it crashes against the stage. Yeah, yeah. And even when I lower the stage, the distance is not right. Right. So that's a little bit one of the problems that I found. Yeah, that this is too close. Right. So the problem is even though it fits physically in here, I'm not able to use that, even though it's still 160 millimeter objective. So what in the world's going on here? Yeah? And the reason is because those two objectives have a different conjugate distance. Yeah, this one is a so-called a short barrel objective, which has a so-called a conjugate distance of 185 millimeters, and these are the standard ones of 195 millimeters. And this old Olympus. The old Olympus objective over here also is a short barrel. So this is the old Olympus system. So I find it interesting that some of the modern microscopes, modern introductory microscopes, that they actually also are using the same system size of objectives like the old Olympus microscope. Yeah. However, I just want to show you something else here, because I do have the old Olympus microscope also next to me. I need to go down here on the floor just a second. It's a little bit heavier. Okay, I'm going to show this to you. Yeah? It's a so-called, it's an Olympus CHA microscope from the 1970s, 1980s. I basically converted it also to phase contrast. You see over here, these are the short barrel objectives as well. But the thing is that the distance now is large enough that I am now also able to insert larger objectives. Okay, so you see that it's kind of semi-compatible. So if I want to, I could upgrade this microscope here with larger objectives. So that's kind of nice. But I cannot upgrade just a second again, the small microscope over here with larger objectives. Yeah. So because even though it physically would fit and probably also optically would fit, there's not enough space down here. Yeah. So this is simply something that I kind of want to inform you, because I do occasionally get questions, and then I say, yeah, you can exchange objectives if you just make sure that it's also 160, and I'm saying, well, maybe not always, because not all 160 millimeter objectives have the same so-called conjugate distance, right? Huh? So this is a little bit of an issue. The good news is I have not seen so many of those small short barrel microscope objectives around. However, when you buy replacement objectives, it's sometimes not indicated. Yeah, and on the picture you sometimes don't see if it's a small objective or a large one. Yeah, so this is something that I kind of would like to simply share with you. That that's also something that you have to take into consideration. Now, to make the issues a little bit more complicated, isn't this beautiful? It's yet more complicated. This introductory microscope here, I said is a little bit better than some of the others because indeed it has those larger 160 millimeter objectives, because there are also cheaper versions of such microscopes around that have even smaller objectives, and they don't even fit, right? They're not even compatible. Yeah, so it's kind of interesting a little bit that of the different standards that there are, and those objectives here I've also seen they give a little bit of slightly better contrast. Can I do a little rant here as well? I'm just gonna tell you a complaint generally about those, and I don't understand why those. I just have to say it. Look, here now you see this. There is this ring here that you can turn. Okay? And what does this ring do? It is so sad. And I don't get it. I don't get it why the microscope manufacturers don't correct for that. Look, here it's the bottom view. Yeah, it's just a plastic disc. Yeah, and you have holes in here of different diameter, and this is supposed to simulate or emulate the aperture diaphragm, right? Normally you have in a condenser, you have this lever that you can move back and forth to change the aperture. And this is basically supposed to emulate this. This would be actually a nice idea, but there's only one problem. It doesn't work because all of the diameters here are too big. It makes no difference. Yeah, so this is a little bit of a pity, and where I don't get it. Come on, yeah. So what you can do is, if you have a microscope like this, and I have tried that, what you do is you take an old credit card or something, a piece of plastic, and you make a tiny hole in it, and you kind of put it over here to actually really decrease the aperture. What you're going to see that, especially with a 40 times objective, indeed you're really going to increase the depth of field. It's going to be significantly darker, but indeed you're actually getting an effect. Okay, so this is a little bit some of the things. Some companies also put some color filters in here, which don't have a big of an effect. So I don't know. These wheels here are not really useful. What I now have tried in one of those microscopes is, well, why not put some polarizing filters over here on top of those? You put some polarizing film on here, and then you basically have polarized light, and then you can put the specimen on here and the second polarizing filter on top. This does not work because the plastic lens that you have in here starts to depolarize everything again. Yeah? So you basically polarize it down here, and then the plastic lens destroys everything again. So yeah, you see, I've been doing around a lot of experimentation with those microscopes, because otherwise they are, let's put it this way, not so bad considering their price. Okay, and the reason why I am still interested in those low-cost microscopes is quite simply because they make very nice travel microscopes. Thank you very much. I got some of the questions now. Thank you. Yeah. And that is actually something. One of the reasons why I'm taking them a little bit serious. Let's put it this way. I'm trying to take them a little bit more serious as well, and trying to tweak them and optimize them, because if I go on a trip, on a travel or so, and I want to do some microscopy, and if I do not want to take along some samples, then I can actually try to take those along here as well. Okay, so just wanted to share that with you as well. Yeah, simply to be that you're a little bit aware of the different types of. I'm not, maybe one of the upcoming streams or so, I might be talking about some infinity optics, which is again a completely different category. Yeah. But in any case, just wanted to show you that those old Olympus objectives basically have the same standard as some of the modern low-cost introductory objectives that you find. Yeah, and of course you probably always want to, but I'm going to do this later on, but just, you always want to make sure that they're properly stored away, dust-free and protected. I'm going to do this later. Okay, so let's put it somewhere over here. Oh yeah, by the way, I've got also some, I was thinking about talking also a little bit about LED exchanging of microscope lamps and so on. So, what do I have next? Let me see. Okay, most of you want to have a look at the vegan meat. Okay, let's give that a try. Okay, so what did I do? Where is the vegan meat? I'll tell you some background story here as well. Several months ago, last year, I forgot. I basically had some lunch somewhere, and they basically said, yeah, it's for, some kind of some dish without actual meat. Yeah, and I was it was in a restaurant, and yes, I actually took one of those pieces along home. I rinsed it because there was some sauce over it, and basically I put it into alcohol. And then I also went to the supermarket and I also bought some vegan meat pieces for cooking. And I also put it into alcohol. And what I have done is because, of course, there's water in there. After a couple of days, I've also exchanged the alcohol and added fresh alcohol. So this is alcohol. It's basically been stored like this for a year now. If we want to observe it under the microscope, it would be always a good idea to rehydrate it. But where is the label? Yes. Yes. I know. Yeah, it's because sometimes I do collect specimens. I don't label them, and then I forgot what it actually is. So what I've done now is I've basically taken some samples here and and put them into water. The water will do the following now. It will kind of remove the alcohol and rehydrate this. And let's try to find something interesting here. Because I know that I've done this already a few years ago, and I found something in there that it's kind of worth discussing a little bit. And I hope I'm able to find it again. Actually, you don't want to find it. Can I complain about something else? Slightly off topic. There are other microscopy YouTube channels out there who go for the spectacular. So basically they put something, whatever they put under the microscope, they see nematode worms and oh my God, no, that's not the problem. Yeah. And so, people, sometimes some folks try to stir fear, right? By saying, oh my God, how disgusting, and they're in meat, there's all this disgusting stuff. No, that's not the point. Yeah, that's not the case. They're not worms all over the place. Okay? Just saying, yeah, because you do have to stay a little bit truthful as well. Yeah? And there are unfortunately some channels out there that try to, go a little bit on the spectacular. Yeah, and I'm not for that. So a very small amount we need. But however, the thing that does disgust me with that a little bit, which did disturb me, are not some kind of weird microbes. No, of course not. But the fact that I actually found microplastics in them, right? And that's actually, I would say, something that it's more interesting. Yeah. So I'm going to just put this away. I don't know if you have to really rip it apart a little bit. Okay. I'm going to use tissue paper to clean off the tweezers. Okay. This still might be a little bit too thick. Yeah. So then another one over here. I'm going to do it like this. Yeah. And I was kind of wondering, why in the world, why is there microplastics in there? This was kind of the thing that I was kind of wondering. And I think the reason is because these are soy products. And so the soybeans are grown, of course, on a field. There is, of course, it's raining. And scientists and researchers, they have found quite plenty of microplastics also in rain. So this is, and those microplastics are actually fibers. So if you have synthetic clothing with synthetic fibers, then this actually is a problem. And a lot of these synthetic fibers, they also wash out. Let's put this away again. They also wash out when you in the washing machine. So this they found that is actually an interesting issue, and those synthetic fibers actually they do not decompose. So I'm not so much worried about cotton and cellulose fibers and these things because they decompose, but those synthetic fibers they sometimes they don't do that. Yeah. So I wonder if this is actually something that we're also able to see. It, like everything, it does take a little bit of patience. So maybe we're not going to be so lucky. Yeah. But yeah, a couple of months ago when I did that, I was actually, yeah, with a little bit of patience, you were able to find all of these things. So this again is an example of it being way too thick. So I'm going to a little more water. So let's maybe also over here. Yes, I know. I'm kind of in here. They're mixed together. I'm aware of this, right? So so let's have a look and let's see. So no guarantee that yeah, we're able to find anything of interest here as always, but that's also part a little bit of the fun, that sometimes you don't always know what to expect. Yeah, I have to wait again a couple of seconds for the whole thing to appear. Okay, and yeah. Yep. So let's go down a little bit. Yeah, these are clusters, but you see many of those. If you see those, yeah, here we go already. Yeah, sometimes there are those thin, regular-looking fibers in there, where I know that this is not plant material. Some of those irregular ones could be plant material. Yeah. But let me see, maybe the other sample is a little bit thinner over here. Let me go down a little bit with the magnification. Let's also remove the polarization here. Yeah. Yeah, for example, those things here, yeah. Yeah. You find a lot of these fiber-like structures, and some, I'm not saying all of them, but some of them could actually be synthetic. And I'm kind of wondering, I mean, yeah, how does this stuff find its way in there? Yeah. I've a couple of months ago, I've also seen stained ones, like blue ones, for example. Yeah, yeah. So this is a little bit something where I'm a little bit. Yeah, which kind of shows how everything is a little bit connected to each other. Yeah. I probably have to break it apart yet further. Let's go down again with the magnification. Yeah, my mistake here is I did not spread it apart enough. Yeah. Sometimes what you see is that those droplets that you see sometimes are not necessarily, this one is an air bubble, but I've also seen some fat droplets in fresh ones, because this one has been in alcohol. Yeah, all of the fat droplets, the fat has been dissolved, of course. Wow, what is this here? This also looks like a trichome. What a nice coincidence. I don't know. Oh, this is definitely a trichome. Now, let's be careful here. Let's think a little bit. You always, as a scientist, you got to be careful. Did I use those tweezers here, maybe? Yeah? Did I use those tweezers to actually pick it out? I mean, this could, of course, be a contaminant from the tool here. Yeah. So yeah. So this is this. Can you mention Nile red to identify plastics? Nile red is a stain. It is a hydrophobic stain. I've not used it yet. There's also a YouTube channel called Nile red, but the Nile red actually is a stain, which is fluorescent. So this means if you stain a sample with Nile red, then and you use UV light, then the microplastics will actually start to shine. So this one here, this trichome, I would not give it too much of a, how do you say, it's not a problem anyway, but I would not give too much emphasis on that. It could indeed be because I did not properly clean my my tweezers. Yeah, but that is essentially, this is how do you say, vegan or soybean-based yeah, meat looks like under the microscope. And what I think I would do is, now polarization is the following. Yes, however, many substances, especially if you have such a mix of different things in here, many of the things start to depolarize. I've seen. So basically what happens, even if you have some other object in there, and if the polarized light jumps to it, it depolarizes it, and you still see it shining on a dark background. Yeah? So this means that even fibers indeed do change the color also around, but sometimes it's I found it sometimes a little bit difficult. Yeah. So what we do is if I use DIC, for example, yeah. So then I actually have let me put this back here. Now I'm using a little bit polarized light, and I can actually change the colors around here. Okay. Yep. Yeah, so again, here in the back, maybe this could be, those string-like structures could be some, yeah. Let's move over here a little bit if there's something over here. Yeah, see, the problem with this sample is it's simply too dense. Yeah, so let's try the other one. Those things here indeed could be either they're bubbles, air bubbles, or could be oil droplets. But I do not think oil droplets because of the alcohol. Yeah. What happens if you add a lambda quarter plate? Then essentially, I do not have regular polarized light here. I actually have a so-called also Wollaston prism because of DIC. It is therefore not. If you know what, let's try, because you're asking. Let's just try normal polarized microscopy. Okay, I have to flip this out. I have to take this out. I put a normal polarization filter here on the bottom. Okay, I put one on top. I'm going to now use the 4 times. So, yep, it's now moving because the filter is touching. I'm now rotating the, and that's basically how it looks like in polarized light. You see, sometimes it starts to shine up some, but let's give it a little bit so that we're actually able to see it better. Yeah. It would be now really interesting to identify what those bright pieces are. Ah, here. That's certainly dust fiber, or yeah, some kind of possible microplastics. Yeah, it's actually quite interesting. You don't see it. You don't see it like this. You turn the polarizing filter and you see it. Okay, so maybe yeah. Again, it could also be if it's a cellulose fiber. Maybe it would also shine up like that. Yeah, but in any case, there are lots of things in there that where I'm kind of wondering what it could be. Yeah. So and you just basically So this is now regular polarized, just like I did before with the trichomes. I don't know what this is. Ah, yeah, also, yeah, something. Yeah. Here, also a few of them. Actually, honestly, this was actually a good recommendation. See, you barely see them. Yeah, on the bright field. All of the things in the back, all of those irregular structures that you see, these are bubbles. That's actually pretty cool. Yeah. Let's leave it a little bit so that we're able to orient ourselves. Yeah. Here another one. Yeah. I think you get the idea. Yeah. I was actually thinking. I wonder what that is. That's the one from before. I was actually thinking even of a possible maybe citizen science project where people, I don't know, do some environmental assessment and finding microplastics or things like that. Yeah. Would salt crystals in the food glow too? First of all, I think I tried that once. Salt crystals are not polarizing, so I don't think so. I've not tried it with regular polarizing filters. However, in food, if you have salt in food, then you have food contains water. Then the crystals would dissolve and you would not see it. So, not so to correct your comment here is would salt crystals in food glow too? Then I said in food not, because food is moist, and this dissolves the crystals. So it's got to be in a dry form. Yeah. Yeah. So what makes something polarizable? Okay, the following situation. If you have, I don't have this here now, but that's maybe, you know what, I'm gonna do this for maybe in a future live stream. If there are, for example, if you've got cellulose tape, and if there are basically, if the fibers, like the polymers, are kind of oriented in a certain way, then they're polarizing. Okay. However, some structures, like we've seen over here, they themselves are not polarizing, but they simply depolarize the light. Right. So basically, the light rays enter the specimen in one plane, then they hit the specimen, and the specimen, if it's polarizing, then it will simply turn the plane. However, some specimens, what they will do is they will simply, the light enters in one plane, but it will come out in all four different directions, and then you're also able to see it as a bright on a dark background. Yeah, so this is a little bit the thing here, and and what we see here is that some of those structures, could either be polarizing by turning the light, and then you see it, or by simply depolarizing it. Here again, a trichome. Yeah, most like possibly maybe from my here again over here. Yeah, so yeah, basically it's something I just wanted to also share with you. Yeah, also as a possible, how do you say, thing to try out. Okay, so now what? Now what? The next one is viewer questions, Q&A. You see, I'm kind of trying to stick to the 15-minute format. And here my son again prepared some questions. He complained a little bit to me today. He said, I'm already answering too many questions as I go along. So he kind of felt a little bit useless putting those questions together. Okay. Okay. What happens if you put DIC back in place? I have no idea. So let me see. So that's not polarization. So what I'm going to do now is I'm going to put in the one on the bottom. Okay, and I'm going to put in the one on the top. It still polarizes. Let's put this one here. Let's put this one here. Brighter. Refocus. Yeah, it looks kind of weird. This is basically DIC and polarized light. Yeah. Kind of destroy. Yeah, indeed, it seems to destroy the effect. Yeah. So that's, Okay, let me quickly, because it's already 1 hour and 18 minutes, let me quickly go through some of your questions. Yeah. Again, I hope that I'm just going to skip over those that I already addressed, okay? Okay, I also talked about this. Make sure whichever channel it's... this is basically what for the time being, what I would like to try is I would like to keep all of this practical hands-on microscopy stuff on this channel here. Maybe you... I would be happy if you could also... I'm going to maybe tomorrow or so upload an explainer video, a more or less traditional explainer video to my other channel, to the main channel, and I would simply want to see how that works. Okay, I'm in an experimentation phase, so maybe it won't work, maybe it will. I don't know how much interest it's going to generate or not. Okay, Yeah, I still talk about, yeah, it's still about I agree YouTube Shorts does mess with the algorithm. Yeah, and I have found out pretty the hard way. Don't worry too much about subscribers, though. It's generally known that the majority of views will be not be subscribers. Yes. Yeah. That's also another thing, is subscribers are not as important as they used to be. However, it does show when people subscribe that I'm a little bit on the right track. And I want to make videos generally that not only interest me. For me, this is mainly a hobby, what I'm doing here. But I, of course, want that other people also benefit. Yeah. I don't always have time for videos. Okay. Trichomes. I go through this. Can insects enter the leaves through the stoma? Can insects? I know that there are some mites that are very small. And I will give you, for this question, I'm going to give you the honest answer is I don't know. I don't know of any insects or mites. However, I'm going to give you now the answer that some of my university professors have given us, and I also give the same answer to my students because I'm a teacher in school. It's kind of a little bit avoiding the problem. If you already ask, can insects enter the leaves through the stoma? If it is conceivable, if you can imagine it, probably in nature somewhere you're going to find it. Okay, so that's kind of the rule of thumb. Yeah, even though I don't know of any, chances are pretty good. If you look hard enough in nature, you're going to find something somewhere. Maybe it's not yet been discovered, but nature is so diverse, and this would not violate any laws of physics or of biology or of chemistry. That's kind of the how do you say, the cutoff. Yeah. If you were to ask, can insects or bacteria survive, I don't know, fire, then I would say no. Yeah, because that would actually break the chemical bonds, and that would actually be a violation of the laws of physics and chemistry, and would be against, life is not sustainable, right? But that is something that is conceivable. That's why you're asking the question. And for this reason, probably in nature somewhere you're going to find it. Okay? Stoma, stomata, stomates, I guess there are different ways here. Yeah? It's a cross polarization here. What you taught us. I know it's almost all the time. It's amazing. Yeah. Is it possible to show DSC and polarization? I have done this right now. More likely lamb's ear by the looks of it. I don't think by the leaves have trichomes. Okay, I don't know. I'm not so much into botany. Yeah. So maybe yeah. I unfortunately I don't know the name of the leaf. Yeah, I tried that, the 3D wire polarization using my other microscope. I did not get a very strong effect, I have to admit. Could be that the sample was not suitable, but I'm still I'm working on it. Okay, so I'm still optimizing it. Would the bacteria stand out in bright field? I tried that with methylene blue, the staining not. The problem with methylene blue or any other stains is that it stains pretty much everything, and that's a little bit. Also the problem, then you have essentially you're killing off the organisms, and then you also do not have any movement, and then it might actually be even more difficult to distinguish them. Yeah. Bacteria are so interesting. Can a bacteria that can interact with Earth's magnetic field? Yes. Yeah. Purple sulfur bacteria, the proteobacteria. Yes, these are magnetotactic bacteria do also exist. Yeah, they're basically who are attracted by magnets. Yeah. So I don't know about any microbiological indicators for heavy metals, but I know that there are, it's fairly, I think, not so difficult to actually make chemical tests. Okay, at least Marie loves the new format. Okay, yeah. What do you think of Lacerta brand microscopes? I don't know them. I will just say that many microscopes these days, especially the low-cost ones, which are not from the big microscope manufacturers, are simply rebranded. And therefore, they're quite similar. Yeah? So, for example, this one over here, this microscope, even though it's branded as a Swift microscope, you're going to find the same one or similar ones, very similar ones, also under different brand names. Yeah, and that's generally the case these days. Yeah. So I would say it depends more on the individual microscope than on the brand. Yeah. Will the quality of the image change with versus or without the black piece at the back? The black piece at the back. I'm a little bit at a loss now. I've lost the context here a little bit. Okay. Fix the vignetting. Okay. Yes. When you had the objectives out. Oh. Quality of the image change with. When you had the objectives out. Are you talking about this one here? This black piece here? Is this the, or are you referring to this? Some of the objectives had a plastic ring in there. Is this? I don't know about this one. I don't know if it, okay, this one over here is not a plastic ring, and this one also here not. I know that some of them actually had a removable plastic cap that you can take out. I've not tried that. I think it can indeed, how do you call this? In telescopes sometimes have, this is called baffles to reduce the internal reflections. Yeah, so I think it could indeed have an impact but I did not try that out, and I know that some objectives actually have this plastic cup in here with a small hole in the center, and I think that this could reduce internal reflections. Yeah. Can you show us what polarization and DIC setup looks like on your microscope? Yeah, I don't know how I'm going to do that. I'd have to swing this over. Thickness needle. Let me quickly read this here. Take out the disc out, put black tape over the holes and new holes in the middle, each with different thickness needles. You can also, yes, that's basically refers to this microscope over here on the bottom. Yeah, where you can change the size here with black tape. That's also a good idea. So the setup looks like this, and now it's kind of upside down. So what I'm going to do is the following. I'm down here. This is the filter that I normally don't use. Up here, I also put this other filter, the second one, pretty improvised on top. It's a little bit wet now, right? So that's the way that it normally looks. And for DIC, I have this. It's upside down now as well. Oh. That's the camera. Yeah, I cannot show this because the camera is. But there is a little. How do you say this? Yeah. Okay. Right above here. Yeah. This one here. When I turn this, I'm able to change the color. Right. And on the bottom there is also a. How do you say? You cannot see that. I can turn this here. Yeah. And this one also changes the polarization. Yeah. So that's a little bit the how the setup looks like. Yeah. So, let me quickly see if there are any other comments for me. Would like to see vitamin C crystals and polarization again. Thank you for the recommendation. I have not done this for a couple of months now, but I'll pick up on that again. Okay, I've talked about this here. Polarizable, what happens if you put DSC back in place? Yeah, I've been actively debunking these fear-mongering microscopy channels. Funny thing, they often use compound microscopes, but then show stereomicroscopic images. Honestly, there is this one channel, and I say, gee, why even bother to make, honestly, maybe I'm being a little too critical. I don't know. Why even bother to make videos if you don't want to do it properly? And there's this one channel, and it does like this. They kind of show you the microscope slide, and they take a sample, I don't know, a huge one. They put it on like this, and they put the whole thing under the microscope, and then they kind of show you, no, you're not going to see anything like this. Yeah. Or worse yet, they do something like this, and then they just slap on a microscope like this, and then basically they put it on. No, that's not how you prepare a specimen. Why even bother? making videos like this if you don't want to show it properly. Maybe I'm being a little too nasty here, but this kind of is, yeah. As someone who's trying to educate, it's kind of a little bit frustrating for me. Yeah, that's just my comment. Yeah, yeah. It's easy to go viral with scary things. People panic and pass it on quickly, then they get sponsorship and earn money. That's not the way that I want to do it in any case. Yeah, I mean, my philosophy is a little bit, I mean, as a biology teacher, I like to, of course, I'm generally interested in nature observation and so on, and in sharing things. And it's a hobby for me primarily. But sometimes when I show things, there is nothing to see really. But I think that's part of the experience, right? Because that's kind of authentic. And I think it's this authenticity. Not everything has to be perfect. I say, and sometimes, yeah, I don't know, when you put stuff under the microscope like those, how do you say those? The meat, yeah, the soybean meat right now, which hopefully will, yeah, then that's basically maybe not super exciting to see, yeah, but at least that's how it looks like, right? And then we can have a discussion on whether those fibers that we see here should be in there or not, okay? But at least it's authentic, yeah, and not some kind of a fabricated thing, yeah. Can you show what polarization and DSC setup looks like? Actually, I thought I just did that. Right, there is a how do you say a condenser on the bottom. Yeah. So again, yeah. So I've got here a condenser. Yeah. Down here I've got a little wheel that I can turn to adjust the orientation of the polarizer, and up here, yeah, which is so difficult to see. Here, this one over here. Yeah. When I turn that, then I'm able to change a prism around, and this changes the colors. Okay, that's a little bit, yeah. And if I don't want to have it anymore, I can actually pull it out. Yeah, and then, yeah. So that's how the DIC setup looks like. Yeah. So people, yeah. I think that's enough for today. For those, just as a summary, that's kind of what I try to cover, about 15 minutes each. This is the new, how do you say, experimental format that I have. Yeah, would like to hear from you what your view is, opinion is about this format. Yeah, I'm going to post another video, a different one, on my main channel, on the Microbehunter channel. So I'm trying to pivot the contents a little bit. Would like to hear from you what your opinion is of that as well. And yeah, I'm just going to leave it at that. So far the feedback seems to be fine. It makes it a little bit easier for me as well, because I've got several smaller topics to talk about. Yeah. And for me, that's it. Check out the other videos that I'm publishing. I'm trying to publish now more regularly as well. I'm going to simply wish you all the best. Happy microbe hunting as always, and hope to see you again next week. Okay, bye-bye.